Our next lesson was on pathogens and disease. The big thing we want to understand is that what bacteria are and how those are different from viruses. So we have just some basic bacteria anatomy here. Okay, inside the bacteria, we're going to have DNA. Bacteria will all have DNA. That DNA is not housed in a nucleus like you would see in a plant or animal cell. So that's a major distinction for bacterial cells. They are going to have ribosomes, organelles that build proteins. Those organelles are essentially what carry out the instructions of the DNA. We're going to have a cell membrane. That's this kind of tan structure here. Okay. And then on the outside of that, we're going to have a cell wall. Cell walls are also found in plant cells and I believe fungal cells, but they're different in terms of their makeup, right? These are going to be made out of cellulose. Plant cells cell walls are made out of cellulose, whereas a bacterial cell wall is made out of peptidoglycan. So it's a different molecules that make that up. And that gives us our basic structure of the cell wall. We have two different categories of bacteria based on some of them having an extra layer. Some of them get this fatty, sugary layer on the outside of that peptidoglycan layer. So they have the plasma membrane, they have the cell wall, and then they have this slimy layer on the outside. Well, one of the ways that we can help identify what species of bacteria is infecting someone is we can stain that with a stain that actually attaches to the cell wall here. And if that happens, that cell wall turns dark purple. If it doesn't attach to the cell wall, that's because this slimy layer is blocking the dye from attaching. And so that can help us identify that. Now, the reason why that's significant is because this slimy layer can actually be one of the toxins that helps a bacteria make you sick, right? But on top of that, it helps with identifying what the type of bacteria is. So it's kind of a key process that was discovered, this Gram staining. So they also can produce toxins from within their cells that can be exported, that can make cells sick and ultimately make you sick. But that's kind of a brief breakdown of those structures, right? And then we come up with the naming systems for these, which that's not going to be a key part of any test or assignment that you're asked to do. But it is kind of nice to know where some of these naming structures come from. And it's due to the kind of describing the way that the bacteria interact with each other. And yeah, so bacteria are treated when they infect us with most often antibiotics. And these are actually typically molecules, proteins that we've discovered by studying other organisms like fungus, who use this as their immune system for fighting off bacteria. And they typically attack the cell wall of the bacteria. And so there are some vaccines for bacteria too that again are going to essentially train your immune system to attack certain aspects of the bacteria. They're less common. Antibiotics is typically how we treat bacteria. And that is going to be really critical that we've continued to develop new antibiotics because these bacteria evolve, right? This is a major evolutionary pressure. And if there's any advantage that exists in the population, it quickly becomes what is prevalent in the strain, and you get antibiotics over time becoming effective because of the evolution of bacteria. The other type of pathogen is going to be viruses, and 3 years ago, I probably had to go into more detail and explain more about viruses, but people, due to the pandemic, have become more and more familiar with them. we are going to have some key aspects of viruses that are different from bacteria. First, they don't have a cell membrane. This is one of the reasons why many scientists do not consider viruses to be alive. The second thing is that viruses are what we call obligate reproducers, meaning they cannot reproduce on their own. They must have a host. And this is what they use us for. Viruses infect their host, they get their genetic material into the cell, and then they use the ribosomes of the cell, the other machinery of the cell, to get that cell to make copies of the virus. So viruses can have DNA, just like bacteria or you, or they can have RNA, which is a very close chemical cousin to DNA, but it's a little bit different. We'll go over some of those differences a little bit later today. But viruses can work off of both of those different viral groups. Will have some will all have DNA, some will all have RNA. Outside of the genetic material, you typically have a protein coat called a capsid, and that's what the one of the things the virus uses your cells to do is use it to build the capsid coat, in addition to making copies of the genetic material. These guys are pretty good at learning how to evade your immune system. So one thing they can do is they can take and wrap themselves in your own cell membrane, right? That's what an enveloped virus is. A non-enveloped virus would just be the protein coat. So I believe coronavirus, for example, is going to have just the protein coat on the outside, and the shape of that protein coat is what gives it that spiky structure that gives it its name. Yeah, so enveloped and non-enveloped, they each can be harmful. It's just a different way of an adaptation to escaping your immune system. So that's a common thing to see in viruses. Now, there's two different ways that viruses can go about infecting you. They can be a lytic virus or a lysogenic virus. A lytic virus is one that kind of your more traditional illness, right? Coronavirus, influenza, they all would infect you this way. They're going to get into you, get into the host cells, get those host cells making new copies of the virus, and they're going to make hundreds, if not thousands. They're going to burst that cell open and then go and infect new cells. That's the way lytic life is. Very short. It's going to be days from infection to illness. Lysogenic viruses work differently. What they do is they actually get their genetic material into the host DNA, and then it lies dormant. And then every time that cell divides, it's going to go through S phase of cell cycle, and it's going to make a copy of that DNA of the virus in addition to its own DNA. And it's going to infect the cells by having the cells make copies of themselves, including the virus DNA. And then a trigger of some kind, whether it be stress, a fever, or just that cell changing in some way, it can trigger that cell to start making copies of the viral DNA or viral RNA. And once it makes copies of it, then it's going to go through and make the capsid coat, and then you have an active infection. This is like when we talk about like a HIV is a classic example of this. HPV is another one that works this way. If you've ever had a cold sore, right, it tends to come back in about the same spot every time. The reason why is because that section of your cells all have a lysogenic virus that infected them, or infected just a few cells, and then they divide over and over again, and they made copies of that all in that same localized area. And then whatever conditions are needed to cause the infection to become active trigger, and then you get the sore that develops. So lysogenic viruses, very difficult to treat, right? How do you know that you've killed all the DNA that's hiding in your own DNA? This is one of the things that makes HIV and AIDS so difficult to treat. So let's see. Yeah, I think that's the main things I wanted to go over from this lesson.